So, good evening, everyone. Today we are going to talk on starting the session on food microbiology, and this particular session will happen in two slots. Today, the first 1 hour is going to be introduction to microbiology. So, I will take you through on the basics of microbiology, and tomorrow we will talk specifically on food microbiology. So, welcome to those who has already joined the webinar, and those who are not able to come yet, I think they are going to miss some refresher course today. So this basically chapter will take you through, refresh your memories about what you have learned in graduation classes and post-graduation classes. So we have tried to include end-to-end possible topics which relates to microbiology basics. Before we start, let me introduce myself. My name is Virendra. I am marketing manager for biomonitoring or microbiology division of Merck in India. I have been working with Merck since 2008. So this is around 11 years since then. I'm postgraduate in microbiology. I am certified trainer for Asia Pacific and also the Nestle Key Account Management for Asia. During the tenure, there are various projects which have been launched in India, and there are quite few programs which relates to leadership or the management that I've gone through while working in Merck. So in case of any queries about microbiology, since our expertise, the expertise of me and my division belongs to microbiology solutions in food and beverage and pharmaceutical industry, you are welcome to ask any questions, or in future, if you want to connect with us, you can write back to me at the email ID, which is given in bold, virendra.asati@merckgroup.com. After this, let's try to see what is the content that we have in Kitty today. So we would like to discuss some basics on binomial nomenclature. So some refreshing ideas for you. We will take you through about Whitaker's 5 kingdoms and Carl Woolf's 3 kingdom classification systems and what is their practical utilities in today's world. Do we really have something to do with, or it is or whether it is only the academia which is working and using these kind of systems of nomenclature. We will also try to understand the difference between prokaryotic and eukaryotic microorganisms. So some more basics on this. There will be some overview on the bacterial size, shape, and arrangement. So how do we classify bacteria based on these parameters? There will be some dedicated slides on cell wall. So cell wall relating to gram-negative, gram-positive bacteria, archibacterial cell wall, difference in gram staining and acid-fast staining and their utilities. We will also talk about some cellular structures like lipopolysaccharides, spheroplasts, protoplasts, and L forms of bacteria. And then a short note on effect of antibiotics and enzymes on the cell wall. We will have some discussion on cell membrane as well. So structure, function, and composition of bacterial cell membrane. And then last but not the least, some review on the reproduction or the growth cycles of the bacteria. So we'll talk about what are the different types of reproduction that these microorganisms follow. What is this growth curve and what are the growth requirements of microorganisms? So I think most of the slides will relate you to your practical works that you are doing on day-to-day basis. So the same kind of refresher course, which can also try to help you understand, probably clarify some more concepts, or may bring you some refreshing uh, breathe to what we are doing today. So let's start with binomial nomenclature. So you know what is binomial nomenclature. You can go through different publications available across the world. This is one of the most acceptable systems across the world on binomial nomenclature. This was the formal system of naming a species of living things by giving each a name composed of two parts. Okay, so every living organism's entity in the world is given a name, and this name contains two parts. The first part is basically is also called as two-term naming system or two-name naming system. So a name consists of two parts, and therefore it is called two-term naming systems or two-name naming system. The first part of the name is the generic name, which identifies the genus or the genus of the organism. And the second part is the species name or the specific epithet, also called as, and it identifies the species. For example, the most popular fruit that we know nowadays, mango, Mangifera indica. So Mangifera is the genus and indica is the species of this plant, which is mango. On the right side, you can see the image of this scientist who actually devised this nomenclature. So what are the rules for binomial nomenclature? The rule, first and foremost, the entire two-part name must be written in italics. So there is no capitals, no smalls only, but there should be italics letter. And on the right side, you have given the same name which is given in different formats. You need to identify which is the correct one based on the rules. Okay. The second one, the genus name is always written first. So the first name which comes, the first part of the name in this system is the genus, and the second part is species. The genus name must be capitalized, which means the first letter of the genus name should be in capital. So for example, if you see in 1, 2, 3, 4, 5 different types of names which is written here, you see which particular naming pattern is following all the rules. And the fourth one, the specific epithet or the species name is never capitalized. So a few rules. Number one, it should be of two names. The first name is genus, second name is species. The whole name should be italics. So you see which one is completely italics. You also see where is the first letter of the genus is into capital and species is not in capital. So if you look at all these rules and regulations, you will find the fifth name Homo sapiens, which is the bottom of all the names, is written rightly based on the based on the binomial nomenclature, and therefore this is the correct way of putting any name based on binomial nomenclature rules. Let's look at another example. Do you see what this flower is? This flower is nothing but Nilambo, the species of aquatic plant with large showy flowers. And Nilambo in very common language, you call it as lotus. Okay. There are multiple species of lotus, and these are very commonly found species across the world. And these are two common names, which is Nilambo lutea and Nilombus lucifera. And you can see this is the way you can differentiate different species. So most of the characteristics of the plant remain same, but they still have some differences in their morphological and genetical compositions also. And therefore it is good to identify them with two species names. One is Nilambo lutea, another is Nilambo lucifera. Commonly all of us call them as lotus. Let's look at different types of classifications which have been prevailing across the world. And if you look at the different kind of publications available, there are multiple ways of classifying the biological kingdom or the biological world. There has been, if you go to history, there has been multiple attempts to classify. So first and foremost attempts, which was uh, thousands of years back, was based on the uses of different biological things. For example, the people used to use some of the things for food, be it animal, be it plant. Few of the things were used for shelter, like animal bark or the plant bigger leaves or the plant barks also, and for the clothing purpose also. So the very primitive kind of classification was based on food, shelter, and clothing, but it was just for the namesake, but it has no really practical or scientific hold behind it. Then came some scientific basis of classifications starting from Aristotle, who actually put in plant based on their morphological appearances into herb, shrub, and tree categories, while he also helped animals based on the categories of animals having with or without red blood cells. So this was again another kind of primitive classification which did not had much of scientific hold, except that the approach was little scientific based on the physical and morphological characters. Then came Linnaeus, Carolus Linnaeus, who is responsible for also for the nomenclature. He actually came with an idea of two kingdom classification, and the two kingdoms were largely classified into Plantae and Animalia. Now, plant and Animalia, again, this was based on morphological or the appearances or the behaviors which are apparent to a person who is seeing them, but nobody went inside into their compositions or the genetical makeup of these varieties of genera. So basically, these systems did not address or distinguish what is eukaryotic and prokaryotic, and this actually classification did not have any idea what eukaryote or what prokaryote is. They also did not talk about unicellular or multicellular organisms, which is basically the different steps of evolution, how multicellular evolved from unicellulars and so on. So this message was not there definitely. There was no talk on photo and non-photosynthetic kind of organisms, and it also failed to establish linkages of evolution between different set of organisms. So this was the old system of classification which has a lot of limitations into this. Nevertheless, there further in more detail, there came with 2 kingdom, 3 kingdom, 4 kingdom classifications. So as I said, like Carl Linnaeus, who gave actually 2 kingdom classification for animals and plants. Then Haeckel in 1866, after 1735 of Carl Linnaeus, Haeckel introduced in 1866 3 kingdom classification, which was protist, plants and animals. And then in 1938, Copeland was actually a very big uh, critical competitor of forthcoming classifications also, which was basically the 5 kingdom classification. So he was a close competitor of that. So he proposed 4 kingdom classification in 1938, which was based on Monera, protists, plants and animals. If you look at the entire classification, age, history and everything, there is an era which is until 1969. If you see the pink two-headed arrow below on the lower side, so there was a time wherein until to 1969 the 2 kingdom classification was accepted. Between 1969 to until 1969, 2, 3, 4 kingdom classification, everything came, but it is the 2 kingdom classification which was mostly accepted. While from 1969 until 1990, which is hardly 3 decades back, the 5 kingdom classification was accepted, which we will talk a little more. And after 1990, it was not in terms of kingdom, but it was in terms of 3 domain. If you see, the terminology have changed. So 1990 onwards, 3 domain classification came into picture, and which is still yet accepted and probably the most advanced system of classification as of now. Let's look at this Whitaker's 5 kingdom classification, which was accepted, which was talked until 1990. And then you see the arrow, so which means we have to read from left to right. So Whitaker's early thinking about kingdom was strongly shaped by his ecological research. So he was basically an ecologist. So most of his idea were relating to ecology of the biodiversity across the globe. But later versions were also heavily influenced by concepts in cell biology. So if you look at the evolution from left to right, if you start from organisms, and then you see the categorization under prokaryotes and eukaryotes. So all the prokaryotes were categorized under Monera. Now see how this 5 kingdom and 3 domains will come in picture. Now eukaryotes actually further divided into unicellular and multicellular. Okay. So if you remember in the slide previously, we said that earlier systems of classification did not talked about prokaryote, eukaryote. They did not talk about unicellular, multicellular, and they did not talk about other cell organizational structures. In this 5 kingdom classification, at least most of the things were included. So you have prokaryotes being called Monera, eukaryote, unicellular being called protist. Then come to multicellular. Multicellular were further into 2 categories, the one with cell wall, the another without cell wall. So animal cells do not have cell wall, while plant cells and fungal, they have cell wall. So with cell wall, you have again heterotrophic and phototropic. Under heterotrophic, you have fungi with the chitinous cell wall. Phototropic plant, which have mostly cellulose cell wall, and those without cell wall were under Animalia kingdom. So these are the 5 kingdoms which was studied, which were part of discussions and publications until from 1969 until 1990. Some example, for example, Monera, you have bacteria, cyanobacteria, and mycoplasma. Some examples of protists, for example, we have diatoms, paramecium, and amoeba. Some examples of fungi, we have yeast, mushrooms, aspergillus, for example, a fungi. In plants, we have all different kind of plants which are there. And in animals, we have human, birds, earthworms, all these examples of animal kingdom. What is the significance of this 5 kingdom classification? So this basically, this type of classification, it served as an important organization scheme for discussing biodiversity. So this is where biodiversity could have been discussed separately for different kingdom, for different set of organisms. It seemed to capture fundamental properties of living kingdoms. As I mentioned earlier, it talked about prokaryote, eukaryote. It talked about different cell types. It talked about unicellular and multicellular. Here the fungi were placed into a separate kingdom as their mode of nutrition differs from others. And also it was able to classify autotrophs and heterotrophs into separate groups. It also very clearly it is visible that 5 kingdom classification is more natural than the 2 kingdom classification for sure. And this system says nothing about how organism within kingdom or between kingdoms may be related to each other. So this is one of the limitations, that it could not establish the evolutionary relationships among the different set of organisms. And this problem was further addressed by three domain systems. So in the bottom of every slide, you can see some footnotes or some references. Those who want to have more details, they can refer those references. So there come the Carl Woolf's three systems classification. And in this system, you can very clearly see three different leaves. Okay, the one, the orange, the peach color, and the green color. One is talking about bacteria, another about archaea, and the third leaf about eukarya. So largely archaea and bacteria are all prokaryotes, while eukarya, as the name suggests, all were eukaryotes. So these are two domains, and in the domains, the every branch can be treated as a kingdom. So it has all five kingdoms included here. And within the kingdoms, if you go down, it's like a set of nomenclature or putting nomenclature. You have sub branches, which gives rise to different group of organisms based on similarity, dissimilarities, and so on. So this is basically a phylogenetic tree. Phylogenetic tree means there is genetic correlation or evolutionary correlation can be established by genetic means. And what are these genetic means? These are based on ribosomal RNA data showing the three domain classification, and all smaller branches can now be considered as a kingdoms here. So this type of classification did not only restore the benefits of five kingdom classification earlier, but this also tried to establish evolutionary relationship between different set of organisms and also separated archaea from other kind of bacteria. Now, what is the significance of this three system classification? This is basically holistic system that contains advantages of five kingdom classification as mentioned. It gives right position to archaea. So archaea, they look like a bacteria, but they have many similarities which can be compared with other set of organisms which closely relates to eukarya also. They can survive in highly inhospitable environments like eukaryotes, which may not be there like for other normal bacteria. So archaebacteria is a set of organisms which looks like bacteria, but actually it was different than bacteria. It emphasizes on genetic similarity over outward appearance and behavior. So it is not on, I mean, the focus was very much on the genetic compositions of the organisms and not on how they look like or how they like behave. So it's more of a genetic relationship establishment in this case. The genetic sequencing has given researchers a whole new way of analyzing relationship between organisms. So it was pretty much better to establish ancestral relationship or who came from where, who was the, let's say, parent of a given genre or species. It was possible to establish using the genetic sequencing methods in this kind of domain system. This also emphasizes the extremely ancient linkages that exist among prokaryotes and protists and the relatively recent relationships of multicellular organisms. So it was both the ways, the oldest of old organisms which are prokaryotes and protists and the newest of all the multicellular organisms. There was possibility of establishing similarities or linkages based on the similarity in the nuclear sequencing. Here is the view. If on the top you can see Monera, Protista, plant, fungi, animalia, this was the five kingdom classification. And the below that the second row which talks about domain bacteria, domain archaea, and domain eukarya. So three domain system. The idea of showing this slide is it actually included everything what we have earlier in five kingdom classification, but it has a better confidence of putting relationship in between by means of RNA sequencing and so on. So it has everything that a researcher or scientist would like to have in terms of having biodiversity or establishing relationship between set of organisms. While doing so, there was a real challenge for the viruses also, which have so far not got any real position into the system. The reason, there is always an ongoing debate whether viruses should be classified, and some arguments that include the fact out of 10 arguments, the key 4 arguments which are mentioned here, they are obligate intercellular parasites that lack metabolism. So they don't have their own system of metabolism. They are obligate intercellular parasite. They means they need a host for their application. So they are not capable of replication outside of a host cell. So they are not living. That's the biggest challenge. So how they can, therefore they can be called as biological. Their placement in the tree would be problematic since it is suspected that viruses have arsenal arisen multiple times, and they have a penchant for harvesting nucleotide sequence from their host, which means they take care of the host machinery. They dominate the host machinery. While they have arisen multiple times, they can be infective at certain stages. They can be ineffective at certain stages when they are outside the host cell. So they are actually problematic case. It is not a very clear-cut case like any other biological organism. And the first argument, which is against, which favors their inclusion actually, it comes from discovery of unusually large and complex viruses, such as mimivirus, that possess typical cellular genes. So in terms of genetic makeup, they have some similarities, but in terms of their metabolism, biochemistry, or their lifestyle, they do not have any similarity to living set of organisms, and therefore the debate is ongoing. And this poor category of I'm not say microorganisms, these particles are yet to find suitable position in the animal kingdom. Now with this, I think we will shift our discussion to the prokaryotes, eukaryotes, and gradually to the content of today's webinar that we have been given in the contents. So let's talk about prokaryote and eukaryote. What prokaryote is, on the right hand side you can see a big image of the bacteria, and as you can see there is inside nucleotide, in bracket we write DNA, and it is clear that there is no nucleus. It is just lying inside the plasma membrane, okay, inside the cytoplasm basically. So what is a prokaryote? A prokaryote is a unicellular organism that lacks a membrane bound nucleus, mitochondria, or any other membrane bound organelles. So none of the organelles, neither the DNA material is bound in a membrane. They are just floating in the pool which is cytoplasm, okay. And they can be divided into like two domains. One is archaea and the bacteria. So like archaebacteria and bacteria, they both are types of prokaryotes. While going to eukaryotes, you can see on the right hand side the image, and this is the beautiful image of a cross section, or let's say it's like a piece of cell which has been removed from here, and you can see what is happening inside. So every structure is well defined. They have got their own like enclosures in which these cell organelles are placed, and nucleus also is inside a nucleoplasm, inside nucleoplore. So how they can be differentiated in terms of statements? The eukaryotes are organisms whose cell have a nucleus enclosed within membrane. They have a well-defined nucleus. And eukaryotic cells also contain other membrane-bound organelles such as mitochondria and the Golgi apparatus. So everything is well defined within their own membranes. In addition, some cells of plants and algae contain chloroplasts, so different organelles. They also have additionally, which helps in the entire cell biology. So this is how eukaryotes are different than prokaryotes in having these different well-defined, well-enclosed structures. Let's see in general what are the differences between prokaryotes and eukaryotes. So in prokaryotes, no membrane-bound nucleus, while in eukaryotes we have it. In prokaryotes, we have peptones, cell wall made up of peptidoglycan, while in eukaryotes, cell wall is made up of chitin and sometimes cellulose also. In prokaryotes, there is no membrane-bound organelle, where in eukaryotes we have membrane-bound organelles. So basically, we call it as compartmentation also. In prokaryotes, we have pili and fimbriae for adhesion and flagella for the rotary movements. Here in case of eukaryotes, we have cilia and flagella, but flagella is mostly used for bending movements and not rotary movements. Okay. We have got mucilaginous capsule in case of prokaryotes, which is formed by the cells. In case of eukaryotes, there is no mucilaginous capsules. The cell size is also different, where in prokaryotes we have 0.5 micron to 100 micron. In case of eukaryotes, it can be from 10 micron to 150 micron. In case of prokaryotes, we have mostly single circular chromosome, and in case of eukaryotes, you have multiple and linear chromosomes. In prokaryotes, mostly they bear only one copy of gene, which means they are haploid. If you remember the chromosomes, they are haploid chromosomes. In case of eukaryotes, they are diploid, which means they have two copies of each genes. Okay. Ribosomes, the most conserved part of any cell structure, they have only 70s ribosomes. In case of eukaryotes, you have 80s and 70s both, so more evolved forms. And in prokaryotes, there are multiple ways of reproduction. The conjugation is one, transduction is also there. So DNA transfer mostly happens by conjugation. In case of eukaryotes, it happens mostly by a process called meiosis. Okay. Now we go to bacterial cell. From the cell types, you go to bacterial cell and the structure and shape, and then we move to cell membrane and then the reproduction. So bacterial cell structure, if you largely see, there are different kind of ingredients that form a cell structure of bacteria. The cell morphology basically will talk about cell shape. Then there is something called cell wall, which includes most of the cell contents. Then there is plasma membrane. we talk about membrane contents. Then there are some extracellular structures which are outside. We are not going to talk in detail. Fimbriae, pili, glycocalyx, flagella for the locomotive purpose, movements or adhesion purpose. Then there are some intracellular structures also which are DNA, plasmids, ribosome, cytoskeleton. But this discussion may not be part of today's session. And then there are some endospores which are adaptive forms or non-vegetative forms of the bacteria that we can talk a bit about it because they're also important. Let's see the cell structure. So the bacterium, despite its simplicity, contains a well-developed cell structure which is responsible for some of its unique biological functions and pathogenicity. Remember that one cell is able to do all the functions that we do as an human being. So we respire, we eat, we excrete, we breathe, and we move. So this one single bacteria can also breathe, can also respire, can do all kind of functions which are required for its survival, including the locomotive functions also. Being simple, it's easy to manipulate. So it's the simplest type of organism. Therefore, for scientists, it is very easy to manipulate, and hence they become ideal tool in biotechnological applications also. So that's why you will see that there are a lot of research which goes on E. coli and other set of organisms for the reasons that they are simple to manipulate. Second, they have got fast generation time. So it is easy to breed them as much as quantity as possible as the scientist wants. Some key components of the cell membrane of a bacteria. It has got cell membrane, the barrier that separates the inside of the cell from the outside. So you can see here it is plasma membrane, the green one. The nucleus or nucleoid. So here there is no nucleus. Basically, it is nucleoid in this case. So this is the location of genetic material. It is just floating inside the cytoplasm. The cytoplasm is basically the swimming pool, the location of the machinery for cell growth and function inside the plasma membrane, and other macromolecules like proteins, nucleic acids, lipids, polysaccharides, etc., which are there inside. So these are all key parts of any bacterial cell structure. Some important components that we would like to discuss is one of them is lipopolysaccharides. So what are lipopolysaccharides? Basically, these are found in the walls of gram-negative bacteria. So going forward, we will also talk about the cell wall structures, and you will find there is something called lipopolysaccharides molecules which are present on the bacterial cell wall. Now, this is also very commonly, they are known as endotoxins, and few of them are also called as lipoglycans. Now, once the endotoxin, the name comes, you can very much relate to the industrial applications of these terms and the concerns which are created by these set of molecules. These are basically toxins released by many of the bacteria. The large molecules consist of a lipid and a polysaccharide composed of a O antigen outer core and the inner core joined by a covalent bond. So if you don't want to read this technical statement, you can look at the left hand side. So there is O antigen on top, then there is the outer core, then there is the inner core, and there is a lipid A. So four components of a lipopolysaccharide which are there. These lipopolysaccharides, they contribute largely to the structural integrity of the bacteria because they form the wall, and wall makes the bacteria, it keeps the bacteria integral, and therefore they contribute a lot to the structural integrity and protects the membrane from certain kinds of chemical attacks. That's what they do. Even though it's endotoxin, it is secreted as part of normal physiological activity of cell's membrane. So those who remember the difference between exotoxin and endotoxin, endotoxins are basically released once the bacterial cell wall ruptures. It is important that the bacteria should be broken down, and therefore whatever released inside should come out. Whereas exotoxin, they are released normally even though the bacteria or the organisms are surviving. Now in this case, it is not true. Although they are called as endotoxin, they can still be released by the live bacteria as a part of physiological activities of the cell membrane. So that's the important part, because the bacteria will survive and the endotoxin will be kept releasing in the body if they infect the human beings. Now the humans are more sensitive to lipopolysaccharides other than animals. This is also true. Lipopolysaccharide act as a prototypical endotoxin because it binds to CD14, TL4, MD2 receptors complex in many cell types. So they have got affinity to the receptors on the different human cell types, and therefore it becomes an ideal point of target to target the human cells. The presence of endotoxin in the blood is called endotoxemia. It can lead to septic shock if the immune response is severely pronounced. So it happens if the care is not taken at the certain period of time, it can lead to a septic shock, and person will have a tough situations if he is not treated as fast as possible. So this is something, one of the molecule of the bacterial cell wall membrane, and has got a lot of relevance to industrial microbiology. Then we talk about three other different forms: spheroplast, protoplast, and L forms. There are the very slight differences. Let's try to understand. So let's look at spheroplast and look at the images also. Left side, you will see the images are little cylindrical, while on the right side, in the gray square, the image is little circular. So what is happening here is spheroplast is a cell from which the peptidoglycan part of cell wall has been removed. So as we said that the bacteria has got peptidoglycan, yeah, in their cell walls. The prokaryotes, they've got peptidoglycans. So once you remove the peptidoglycan, that means cell wall is disrupted. The bacteria automatically, the remaining bacteria will adopt a circular pattern, a circular shape. Okay. So and these circular shape without peptidoglycan, they are called as spheroplast. Now, what is the role? Spheroplast play key role in applications like discovery of antibiotics, patch clamping, cell lysis, and transfection. Please remember, in the forthcoming slide, we will discuss about how the antibiotics act. They act on the cell wall. So once the cell wall is broken, the rest of the material will come out. So the faster the spheroplast formation is, means the new molecule of antibiotic is more effective. So this is the way it comes into application. The formation of spheroplast is indicative of the effectiveness of antibiotics, patch clamping, cell lysis, transfection, and some more studies. So this is one of the applications of spheroplast, very important applications. Then comes protoplast. So in the protoplast, it is not only peptidoglycan, but the entire cell wall is removed. So once the entire cell wall is removed, the complete cytoplasm comes floating out. Okay. So protoplasts are widely used for studying membrane biology, DNA transformation, generation of whole plants, and plant breeding. So if some researcher want to study the action of membrane, they have to actually remove the complete cell wall, and they have to only see the entire cell with membrane only, and therefore they will produce protoplast. There are a lot of protoplast fusion studies also done for biotechnological implications or applications, wherein by fusing the protoplast, the genetic material of the two different somatic cells also is bring together through various means in the lab. So these are the applications of protoplast. And the third comes the L forms bacteria. They are also called as L phase variants basically. So they are cell wall deficient bacteria, but it happens naturally many times because of certain atmospheric conditions or stresses. There is no cell wall, and the bacteria takes a different kind of shape which is called as L shape. Okay, so these are L-shaped bacteria, but they are not induced, but it happens naturally because of certain conditions, because of certain deficiencies in the surroundings. So these are L-form bacteria, and they are used to study early form of life and as host for recombinant protein production also. So if you want to introduce a recombinant protein, these can be a good host because they do not have cell wall and is easy to penetrate a cell membrane than a cell wall. Let's talk about archibacterial cell wall. So we talked both the cell walls, the archibacterial and the bacterial. This is the first one. In the archibacteria, there are different groups of bacteria. You can see on the names: Sulfolobus, you have Ignicoccus hospitalis, then you have Methanosphaera, then you have Methanothermus, then you have Methanospirillum and Methanosarcina. So by the name, you can very clearly make out that these bacteria have got high tolerance to or high tolerance to inhospitable environments. They can survive in extremely acidophilic or temperature conditions. They produce sulfurs, they produce methane gases also, and therefore they have been classified under archibacteria group. And you can look at their cell wall structure. So their cell walls are basically composed of different polysaccharides and proteins with no peptidoglycan. I think this is one of the very important differences, that archibacteria do not have peptidoglycan, while the other bacteria, they have peptidoglycan. Now many archibacteria have cell walls made up of polysaccharide pseudomurine. So it is not true murine, it is pseudomurine, and it is similar to gram positive but lack periplasmic space. So they do not have periplasmic space, which is the space between cell wall and the cell membrane, unlike gram positive bacteria. So this is about archibacterial cell wall. If you go to the other bacterial cell wall, you can see the structures, and they are largely two different kind of cell walls, gram positive and gram negative. In the gram positive, the major difference, if you see, this is the big brick we see in the top, which is basically a thick layer of peptidoglycan, which is there in case of gram positives. While in gram negative, this peptidoglycan layer is very, very thin. Another key difference which you see, the phospholipids on the right hand side on the gram negative, which are actually embedded inside the cell wall and going inside, also creating a kind of passage from cell wall to cell membrane. Phospholipids are present in gram negative, while they are not that much present in gram positive. So we will see how this kind of arrangement or how this kind of composition of the cell wall of a bacteria actually forms the basis of gram staining. Okay. So this, here we talk about gram staining. Gram staining basically was invented by scientist Christian Gram, who was actually working in the morgue. He was working in the morgue of the city hospital in Berlin in 1884. So while working in morgue, he actually created something which has become a milestone in the microbiology, industrial microbiology as of now. It This technique actually differentiates bacteria by cell walls and basically due to because of peptidoglycan composition. The gram-positive bacteria retain the crystal violet dye and thus are stained violet. So let's see on the right side on the images. We have gram-positive on the left-hand side and gram-negative on the right-hand side. Remember, the gram-positive have got thick layer of peptidoglycan. The gram-negative has got very thin layer of the peptidoglycan. So we are first of fixing by means of heat, then we are adding crystal violet stain. Now crystal violet will actually going to stain the entire set of microorganisms which are gram-positive, gram-negative, everything. Then we put iodine. So you remember the iodine complex when we were making stains in our laboratory, crystal violet iodine complex. So they are put together actually, and then we are doing decolorization. So decolorization used to be done with some kind of alcohols. Now remember, these alcohols are basically solvents, and they are going to dissolve anything, the possible lipids, the phospholipids, or the thin peptidoglycan also. What does it mean when you are decolorization on the gram negative, when the peptidoglycan is very thin or there are phospholipids also, they will get dissolved, which means there will be porosity is going to be created. And once the porosity is created, the entire crystal violet iodine will be removed. And therefore, in case of gram negative, it becomes colorless because of the porosity and the removal of crystal violet. And when you put counter stain, they will get counter stain by pink, which is the safranin. So this is the principle. While in case of gram positive, the wall is thick, it is not dissolved by in the decolorization step, and therefore even after doing the counter staining also, the purple color is retained by gram positive. So gram positive look purple because of the thick peptidoglycan layer which is not dissolved. In case of gram negative, they appear pink because the peptidoglycan phospholipids get dissolved and the original crystal violet moves out and the entire cells are counter stained with the safranin and they appear lighter pink in color. So this is a principle which actually talks about the structure and the compositions of the two different cell walls. Now look at acid fast staining. There are still some set of bacteria which cannot be stained through gram positive staining for some of the compositions of their cell walls. And therefore the second type of stain is acid fast stain. This was produced by Paul Ehrlich, also known as Zeal-Nelson staining. And this acid fast staining you will mostly hear in mycobacterial labs, the labs which are dealing in mycobacteria. And the reason mycobacteria, which is basically pathogen for tuberculosis, Mycobacterium tuberculosis, they contain large amount of lipid substances within their cell walls called mycolic acids. Now these mycolic acids, they are not stained by ordinary stains like gram stains. And therefore there was second set of staining system was developed. And these stains are called as Zeal-Nelson stains. They can be basically three set of staining, which is carbolfuscin, alcohol, and methylene blue, three steps to it. And acid fast bacilli are basically bright red after staining, while those who are not acid fast, they will become bluish or dark purple because of methylene blue. And this is because of the similar kind of process that we have talked in gram staining. So you have acid fast, which means high lipid content in the cell wall. So you add carbolfuscin, they get the stain, then you add acid alcohol to decolorize it. So wherever there is no lipid content, they will get dissolved. The cell wall will be porous and therefore the first stain will move out. While in case of acid fast organisms, because of the thick cell wall, it is not going to become porous and the original color will be retained. Now once you counter stain with methylene blue, the bacteria which are non-acid fast, which has become porous, they will get the counter stain. While in case of acid fast organisms, since there is completely blocked, they will retain the original color and they will look like pink. So this is the acid fast staining mechanism. If you see both kind of stainings in a one go separately, it is kind of opposite behavior. In case of gram positive, the purple are gram positive and pink are gram negative. While in case of acid fast staining, it's opposite. Acid fast are basically pink and non-acid fast are mostly purple or blue kind of appearances. So this also will clearly tell you about the differences on two different kind of staining systems and depending upon the target organism that you want to see in the slide. Based on the cell shapes, there are different cell shapes starting from bacillus. Bacillus are rod-shaped bacteria, and if you remember, like different kind of organisms by nomenclature, also E. coli is also rod-shaped bacteria. Bacillus thuringiensis is one of the bacteria which is rod-shaped. Coccus, Streptococcus, Staphylococcus, again different kind of bacteria because they are spherical in shape. Then spirillum, you have different kind of spirillum bacteria also. Spirochete, which are cork screw shaped. Vibrio cholerae, Vibrio parahaematis, which are comma shaped. Chain of cocci, again streptococci, staphylococci, they form chains. Cluster of coccus, so can also be formed. Then pair of cocci like diplococci, you remember Diplococcus pneumonia is the diplococci, and the chain of bacilli, which is again streptobacillus and so on. So based on cell shapes, the nomenclature are given. Mostly the genus name in many of the cases comes from their cell shape, the way the organisms are found in the nature. Now we talked about the organisms, we talked about different kind of structures they form, we talked about their cell walls. Let's also talk about how or the antibiotics act on the cell wall. So this is little complicated. We will just spend some time on this. On the right hand side, you will see a plate, and we call what we see is the basically zone of inhibition. So there is some antibiotic, VA stands for vancomycin, which is kept on the plate, and there is a lawn of bacteria. So if you remember MRSA, methicillin resistant Staph aureus, which has been plated on the plate, and vancomycin showing the zone of inhibition to this, which means it is not allowing bacteria to grow to the level where it has already gone into the plate because of the diffusion into the plate. Okay, so how it is stopping the growth, how it is stopping bacteria to grow, this is there is also chemistry into it. So specific antibacterials interfere with the synthesis of the cell wall. So one thing is very clear, the antibacterials, they interfere with the synthesis of the cell wall. How do they do it? There are different ways. They weaken the peptidoglycan scaffold within the bacterial wall so that the structural integrity eventually fails. They do not allow a peptidoglycan to form, and therefore those structures of walls are not created. Remember, humans do not need peptidoglycans. What does it mean? It means that if you give those antibiotics to humans, it will not hamper the formation of human cell wall, but it is going to hamper the formation of the targets or the pathogen cell wall. So that's how the research and development in the pharmaceutical industry, they choose the antibacterials. Okay. Another example, penicillin, one of the first antibiotics to be used widely, prevents the final cross-linking step or transpeptidation. So in the cell wall, if to form the peptidoglycan, you have to also connect different peptides together, which is the process called transpeptidation. So here the penicillin doesn't allow this to happen, and therefore also the cell wall is not formed. One of the important steps is missed out while cell wall formation, and cell wall is not formed. The third one, the bacteria can also be selectively eradicated by targeting their metabolic pathways and inhibiting protein synthesis by adhering 30S ribosome, thus blocking key RNA interaction. Now, to the entire basis of formation of new life is starts from RNA interaction. And if you remember the transcription step of protein synthesis, RNA plays very key role in that. So what happens? These antibiotics, they adhere to 30S ribosomes, which is part of transcription, and they do not allow transcription to happen, and therefore protein synthesis also do not happen, and this also does not allow bacteria to form, bacterial life to come into existence. So these are the different ways. Let's see an example in this protein synthesis blockage. You can see different antibiotics. You can see like here this chloramphenicol, you can see here in macrolides, clindamycin, streptogramins, you can see aminoglycosides, sites, you can see tetracyclines, and you can see linzoids. So all these kind of antibacterials, they are acting at different stages of transcriptions like the transcription of message of formation of proteins, and by doing so they actually do not allow bacteria to synthesize proteins and therefore new bacteria to born. So there are two ways they can hamper the cell wall synthesis, they can block the protein synthesis, and these are the ways where antibacterial they affect this. Okay. Now, actions of enzymes on the wall. So other than antibacterial, the enzymes also have some effects. Okay. And how these enzymes effects, like we normally use many times enzymes into the food products. Okay. So enzymes that digest peptidoglycans of bacteria collectively called murine hydrolases. Okay. Those which digest peptidoglycans called murine hydrolases. Based on their bond specificity, they are classified as glycosidases, endopeptidases, and amidases. What is glycosidase will do? They split polysaccharide chains. So I will tell you on the right hand side how this happens here. Endopeptidases, they will split polypeptide chains. Again, we will discuss this. Amidases, they basically cleave junction between polysaccharides and polypeptides. So you can see on the right hand side the image, there is on top this NAG, NAM, different kind of sugars there. So this is basically your polysaccharide chain. Okay. So these chains are not formed because of glycosidases. Okay, from left to right. Now the green chains, these are polypeptide chains. So there is something, these endopeptidases, they will not allow these green chains to form. Now there is a connection between polysaccharide and polypeptide, for example, this area. Now this will be interrupted by amidases. So three enzymes, three different functions, three different target locations. This will not allow this to happen, and therefore they will not allow life to born. They will not allow bacteria to grow. So this is the way the enzymes can act in the protein synthesis part or the formation of peptidoglycan cell, which is part of cell wall. The cell wall lytic enzymes are valuable tools for the biotechnologists. So remember, these enzymes are doing lysis of the cell wall. How they can help? They are important for biotechnologists with many applications in medicines, the food industry, and agriculture, and for recovering of intracellular product from yeast or bacteria. So sometimes there are some important metabolites which are produced by microorganisms. So one has to break the cell wall to make sure that we recover the metabolites which are produced inside. So biotechnologists, they will find some good enzymes which are very specific for a particular bacteria type to break them and let the metabolite come out which is of interest for us. So these are the industrial applications. Now we come to cell membrane structure and function. So from cell wall structure, effect of antibiotics and enzymes, we move to cell membranes. Structure and function is, we first take the left hand side. The plasma membrane, also called a cytoplasmic membrane, is the most dynamic structure of the prokaryotic cell. So remember, something which is fluid, okay, something in the fluid phase, it's not a solid structure which has got a skeleton, and it's kind of a floating structure. Therefore it is also called as fluid mosaic model, because it consists of 40% phospholipids and 60% of proteins. So high concentration of proteins and less than half is phospholipids. The phospholipids are amphiphilic, okay, which means both the sides, they have heads which love water, amphiphilic, with a polar hydrophilic glycerol head attached via an ester bond to two non-polar hydrophobic fatty acid tails, which normally forms a bilayer in aqueous environment. So we will see this in the image in the next slide. Dispersed within bilayers are various structural and enzymatic proteins. So you remember that there is a lipid layer in between, phospholipid layer, which has got some amphiphilic or hydrophobic fatty acids, fatty acid heads or tails inside, and between those phospholipids there are certain structural proteins embedded into it. Okay, and now these are floating structures which includes lipids and proteins, and therefore these floating structures are called as fluid mosaic model, the model which was proposed. The function of the membrane, basically they have got a lot of osmotic functions to do. They act as a permeability barrier, they maintain osmotic balance in and outside the cell. So if you remember, if any bacteria is placed inside a, let's say, isotonic solution, it is good. You remember the preparation of diluent inside the laboratory when you are making the dilutions of the bacteria. It is recommended to have a osmotonic solution with a certain molarity. Otherwise, the bacteria will die. Similarly, in the transport medium, similarly the pH of different media, they are given some specific pH so that osmolarity of the diluents of the media should help the growth of bacteria. Okay, otherwise the bacteria will die. The transport system for specific solutes, inside outside transport of nutrients across the cell wall. They also energy generating functions involving respiratory and photosynthetic electron transport systems, establishment of proton motive forces and transmembranes. There are also ATP synthesis, ATPases which are present on the cell membrane. They also help in synthesis of membrane lipids, murin, the cell wall peptidoglycans. They also help in assembly and secretion of extracytoplasmic protein which are thrown outside the cell wall. They also help in coordination of DNA replication and segregation with septum formation and cell division. So if you remember meiosis, mitosis, these membranes also collapse and they divide actually. Chemotaxic movements, the movement of because of touch which happens or because of some chemical induction, they are also responsible for those activities, and the location of specific enzyme systems inside the cell. They perform all these important functions so that a bacteria or an organism can survive. Look at the structure as we talked about fluid mosaic model. You can see the lipid bilayer, the hydrophilic and hydrophobic ends which are there. Okay, so inside you have phospholipid, you have a two tails also, and then you have some proteins which are embedded inside. Okay, so this structure looks like a floating structure. This is not a stable structure, and therefore it is called a fluid mosaic model. You can see inside and outside of the cell membrane. There are two different locations which are shown here, which some proteins, carbohydrate chains, and phospholipids are already there. So now in the last, we are left with last 10 minutes, we talk about microbial growth and reproduction. The microbial growth and reproduction, we'll first talk about the sigmoid growth curve. All of you should be very familiar with this particular graph. This is also called as S growth curve, sigmoid growth curve, or S-shaped growth curve. Okay. So you can see these are the different stages of life cycle of a microorganisms starting at time 0 and the lag phase. Consider when you are inoculating a broth medium or a plate with a loop full of culture. Okay. Now initially you won't see anything. Initially 4 or 5 hours you will not see anything. So that is the lag phase, basically when the culture is trying to adapt to new condition on the plate or in the broth media. This is called lag phase. Then comes the exponential growth phase, wherein the bacteria start multiplying. They have already adapted to the conditions. They have got optimum temperature for growth. They have got optimum pH for growth. They have got optimum water content inside the medium. They have got optimum nutrient inside the medium. They are healthy enough, prepared enough to grow, and there is exponential growth happening here. Then comes the stationary phase, which is on top. Now the stationary phase, you don't see the bacterial count increasing. The reason being, here the number of cells appearing in the medium are equal to the number of cells dying, which means the count remains stagnant here, and therefore it is called a stationary phase. Then comes the death phase. Now in the death phase, the turbidity will slightly decrease. The reason is cells are dying, the walls are breaking, the medium becoming little more transparent. And why it happens? Because of the secondary metabolite production by the bacteria itself, the nutrition is depleting, and because of the toxins accumulating, the other bacteria also start dying, and therefore the death phase comes here. So this is why, I mean, it is said that if there would have not been any kind of death phase, stationary phase today, the bacteria will occupy the entire earth within 24 hours. Okay, so this is the beautiful mechanism which is placed by nature in place, wherein the bacteria cannot overgrow, and after a certain growth they will automatically start dying by themselves. What is generation time? So this is the specific time interval or between two subsequent binary fissions known as the generation time. We also call it as doubling time. So if you look at this on the right hand side, initial, let's say, count was 2. Okay, so bacteria adapted to certain situation, and then doubling happens. 2 2 ja 4, then again doubling, 4 2 ja 8, and then again doubling, 4 2 ja 16. So this is way how the doubling will happens. And now at what time this doubling is happening? For example, let's say first 15 minutes first doubling time, second 15 minutes second doubling time, another 15 minutes third doubling time. So for some bacteria, doubling time can be as good as let's say in minutes. For here you can see the table. Bacillus thermophilus only 1.3 minutes, E. coli it is 20 minutes, and Bacillus subtilis 27 minutes. So that is the reason you will find, while for Bacillus thermophilus you have to have some specific conditions to grow them. E. coli is very easy to grow, and because of the very short doubling time, which is 20 minutes, it is used for many research applications and the activities inside the research or academia laboratories also. Some of the bacterium, for example, Mycobacterium tuberculosis, it takes 792 to 932 minutes. So if you go to any diagnostic lab, you will find that the culture results for Mycobacterium tuberculosis or antibiotic sensitivity testing results for Mycobacterium tuberculosis, they take weeks to months time because of this slow doubling time. Okay, so the entire industrial applications of different kind of diagnostic procedures, or let's say testing procedures or industrial application in food and beverage microbiology, they are largely implicated because of this science behind the bacterial growth. Reproduction. So images speaks everything. There are two modes of reproduction in organisms, asexual and sexual. So in asexual, the bacterial divide by binary fission. So you have all the images here. You can see there. And then there are alternative means, which are budding, conidio, spores, and fragmentation. So binary fission, a cell gradually divides into two. The nuclear material is also divided into two. So this is the binary fission. Then in the budding, one part of the cell protrudes outside with some cytoplasm, and the nucleic material also coming one side. So it becomes second cell, and on the top of second there will be third, and then fourth, and so on. So this way a chain of buds will form, and they will keep on separating one by one. In case of one fragmentation, the entire cytoplasm is actually come with many compartments inside. Okay, the compartments will form like this, and in the big, this happens many times in case of fungi also with the filamentous structures. And because of compartmentation, the fragmentation of one particular fragment will happen in smaller ones, and every small cell will give rise to a new filament. So this is fragmentation. Then comes conjugation, transduction, and transformation, which is part of sexual reproduction of bacteria. So conjugation will happen between two different bacteria. There will be a conjugation tube will be formed. You can see here. Okay. Once the conjugation tube is formed, the genetic material from one is moving to other, and then they will integrate. And therefore, the genetic transfer will happen in this way. In case of transformation, basically a phage, which is a virus, it will transform its genetic material into bacteria, which will then take care of host machinery. So that is transformation. And then in case of transduction, one bacterial cell, the entire bacterial chromosome will be transferred to other bacteria without formation of any kind of tube. Okay, so it is transduced from one to other without formation of tube here. So three different ways of sexual reproduction in case of microorganisms, which are shown here pictorially. Then if they have to grow, they need something for their growth, and these are the growth requirements. So growth requirements for organisms, we talk about temperature, and therefore you have different kind of incubators. They need pH level, and your microbiological medias, diluents, buffer, they talk about this. Oxygen survival, and therefore you have aerobic and anaerobic different way of culturing. Energy to multiply, moisture, and therefore you keep water inside this, and time to multiply, and therefore you say it should give you growth in 18 to 24 hours, or it depends on the bacteria. If you talk about temperature on the right hand side, you will see a temperature scale. The bacteria can survive in very extremely inhospitable environments. It can be as low as -degrees, and then it can be as high as to 110 degrees. So based on their adaptation to different temperature conditions, they can be either psychrophilic, they can be mesophilic. We mostly study, remember, mesophilic bacteria. Then there can be thermophilic, extreme thermophilic, or hyperthermophilic. So most of our laboratory trials of quality control, all they restrict around this mesophilic zones. Okay, we hardly take care of psychrophilic and those, because probably our environments, they themselves will not allow growth of other set of organisms. Another set of requirement of growth is pH. So based on pH requirements also, bacteria can be classified as acidophiles, neutrophiles, and alkaliphiles. Okay, and therefore based on the pH requirements also, there will be different media types. In case of brewery industry or wine industry, we also have some media which are used for which favor the growth of acidic bacteria, sometimes in juice, for juices also. And mostly we have neutrophilic, we have medias to grow neutrophilic bacteria, which grow within a pH range of close to 4. And then we have some alkalophiles. Looking at oxygen demand, they can be aerobic, anaerobic, or facultative anaerobic, which means aerobic, on the top of the media you will have some air here, which is oxygen. And therefore if you put a mixture of bacteria which are aerobes, anaerobes all put together, or facultative anaerobes, the aerobes will grow in top because they need oxygen. Now, as deeper you go inside the medium, because of the high viscosity of the medium broth, sometimes it won't allow oxygen to enter into deep, and therefore all anaerobes will find favorable environment to grow in the bottom of the tube. And then comes facultative anaerobes, which can survive in both the conditions, and they can be found dispersed across the medium. So this is the way, by looking at picture, you can also see that what kind of bacteria is there or might be multiplying inside the culture tube. Based on the energy requirements, they can be categorized as autotrophs and heterotrophs. Autotrophs basically make their own food using simple inorganic carbon sources. Okay, so photoautotrophs, they take energy from sunlight at carbon from carbon dioxide, while chemoautotrophs, they use inorganic compounds as carbon and energy source, for example, hydrogen sulfide. In case of heterotrophs, they require organic carbon sources. So photoheterotrophs, they use light as energy and organic molecules and carbon source, while chemoheterotrophs, they will use organic molecules as energy and carbon sources from chemical sources. I'm sorry for this statement is incomplete. So this is all about today's session. I think we did exactly in 1 hour. Friends, I strongly believe that this is teaching is the highest form of understanding also. So while preparing this kind of information, it was a good situation for me also, that I was also trying to refresh my knowledge of the informations of basics and microbiology. So I thank you all for your patient hearing, and thank you AOSC, thank you Kavita for this opportunity. And I think tomorrow we'll have one session on food microbiology. In case you have time and some questions which I can answer, I should be able to take them. If not, you can write them to the email ID, which is virendra.india@margroup.com.